Hello, welcome to 13 Universal Learning Beyond Barriers. This is cell structure part 2, okay, content specific class, and with you today is Shedro. So I'm going to be taking you around the part 2 of cell structure, okay? So of course, this is cell structure. So if you look at this, this is a cell, this is also a cell, that is a cell. Okay, so let's go like we have always done. What is a cell? What is a cell? You know what is a cell? A cell is the basic or fundamental, okay, this is a cell, basic or fundamental structural and functional unit of life. So you study life at cellular basis. So if you look at these, okay, the structures in cells, these cell organelles and cell structures. All right, you can see them here, all these tiny structures inside the cell, the tiny structures inside the cell. Okay, and if you check, we have these are three different cells. So my question is, what are these cells? Okay, this is plant cell, this is animal cell, this is bacterial cells. Okay, two of these, this and this, they are eukaryotic cells because they have membrane bound, of course, organelles, and they have you can see that this is nuclear membrane enclosing DNA. Same here, nuclear membrane enclosing DNA, but this cell has of course cytoplasmic DNA, right, lying parallel, lying all right, in the cytoplasm in a region called nucleoid. Remember that they all have three things in common: cytoplasm, cell. All cells have cytoplasm, cell membrane and ribosomes. Okay, so let's get into the game, guys. So this is a cell. You can see that this is what type of cell is this? Of course, this is an animal cell. You can see this. This is so guys, yeah, that's the nucleus. The entire thing is nucleus. You're going to discuss it. You can see that nucleus, and this is nucleolus. All right, you see the nuclear membrane is continuous. This is nuclear membrane continuous with rough endoplasmic reticulum. You can see the system of enclosed membranes. Okay, the system of membranes enclosing inner cavity or surrounding an enclosed inner cavity called, all right, rough endoplasm. Now it's rough because it has ribosomes on the outer membrane. Okay, you can see that nuclear membrane is continuous with rough endoplasmic reticulum. Now later you're going to find out that rough endoplasmic reticulum is continuous with Golgi. Golgi, you can see this Golgi, right? You see that, you see these are membrane sacs, okay, that are arranged as flattened stack. All right, okay, you can see this Golgi is continuous with lysosome. All right, so that's centrosome, forms central, right? These are mitochondria, the powerhouse. The mitochondrion is the powerhouse of the cells. This is vacuole, storage, and all of that. That cell membrane, the boundary, forms a barrier. Okay, so you can see we're starting with cell surface membrane. You can see that 7 nanometers. A selectively permeable membrane in plant and animal cells that allows for the exchange of certain biological molecules and ions. Extremely thin with trilaminar appearance. It is comprised of phospholipid bilayers, so made of which are assembled with hydrophilic phosphate heads facing the aqueous environment. The aqueous is outside and inside, okay, and then inside and outside, and the hydrophobic tails facing each other. So that's the barrier. It helps to form a barrier, it helps in cell signaling. All right, it's about 7 nanometer. Let's go on, guys. See it, the um the the cell surface membrane. You can see that. See it here. We have, is made up of phospholipid and proteins. Okay, this is the phosphate head facing towards water. All right, hydrophobic tails withdrawn. We're going to deal with this in topic 4. So you can see the function: barrier between cytoplasm and external environment, cell signaling, cell cognition, okay, cell to cell adhesion, site for enzyme catalyzed reaction, anchoring, cytoskeleton, selection of substances that enter and leave the cell, formation of hydrogen bonds with water for stability. So we're going to discuss it in another topic. Remember that this is acting as a barrier, okay? It's partial. This guy is partial. He allows non-polar molecules to pass through. He hates charged molecules. Okay, let's go to the main organelle. Now this is nucleus, guys. The nucleus is the main organelle, right? So remember, as we go through the cell organelles, we discuss the structure, all right, and of course the function. So if you look at nucleus, nucleus has double membrane. Now there are three organelles that have double membrane. One is nucleus, the other one is chloroplast and mitochondrion. All right now we're discussing nucleus. So you can see the nuclear membrane. Where is it? Nuclear membrane is an envelope, two membrane. Okay, now the nucleus is a double membrane interspersed with nuclear pores. You can see nuclear pores. This is a pore, the nuclear pore. So it is interspersed with nuclear pore. You can see that. So the nuclear pores allows the entry and exit of materials. It allows entry of materials like nucleotides, which will be used as building blocks to build DNA and RNA here. Right, it allows enzymes to come in, ATP to power the process. Okay, it also allows the exit of material like ribosomes. Okay, can exit, mRNA can exit. Okay, you can see the nucleus consists of nuclear membrane. The nuclear membrane, you can see, is continuous with endoplasmic reticulum, especially rough. Okay, it's continuous with that. But if you look at that, okay, let's talk about this. Okay, this part here is called nucleoplasm. Yeah, so you can see nucleoplasm. Nucleoplasm consists of all of these chromatin materials. Okay, which is made up of chromatin material is made up of DNA and histone proteins. Okay, the DNA, one DNA molecule for every 8 histone protein. So the histone proteins help to make DNA compact. All right. So the chromatin condenses to form chromosomes. Don't forget. Okay. That also we have the nucleolus. Nucleolus is deeply stained. Okay, and then it has the highest concentration of chromatin materials. Is that okay? So it stains deeply. It is the site for ribosome synthesis. Okay, guys, don't forget that nucleus controls all nuclear activities because it contains genetic information, contains DNA. All right, so it's the site for transcription. It's the site where DNA replication occurs. So it's an important organelle. So let's do knowledge check. Look at this. It said the electron micrograph shows onion root cells prepared using a freeze fracture technique. The cells were quickly frozen and then physically broken apart. Freeze fractured breaks apart cells along weak areas such as membranes and surfaces of organelles. You can see that. Okay, this is membrane. All right, now so which statement best describes the appearance of the electron micrograph? Now pause the video and just check your knowledge. Okay, try to see if you can solve it. Pause the video. So let's go. Option A says the cells were broken apart at the endoplasmic reticulum. Structure X is a ribosome. Well, does this look like endoplasmic reticulum? Endoplasmic reticulum usually are membranes, all right, which surround an enclosed inner cavity, right? But if you check here, this doesn't look like endoplasmic reticulum. Now we say the cells. Now look at this. This is membranes interspersed with pores, so it's not endoplasmic reticulum. B says the cells were broken apart at nuclear envelope. Structure X is a nuclear pore. Yes, correct. B is correct. Okay, because this structure X is pores that allows the exit and entry of materials. So C says the cells were broken apart at the nuclear envelope. Structure X is a ribosome. No, structure X is actually nuclear pores. All right. Yeah, which allows entry. Now the cells were broken apart at the tonoplast. Structure X, the plasmodium matter. Now this is not vacuole. Vacuole is the membrane of vacuole is tonoplast. Okay, this is nuclear membrane with nuclear pores. All right, so the answer actually is bravo B. option B is the answer for this. Is that okay? So that's the answer. All right, let's go to the next. Now, just to elaborate what we've been talking about nucleus. So you can see nucleus is the size is 10 micrometers, the largest organelle surrounded by a double membrane nuclear envelope and is continuous with rough. See, the nuclear membrane is continuous with rough endoplasmic reticulum. Nuclear pores gaps in the nuclear envelope that allows exchange between the nucleus and cytoplasm. Substances leaving mRNA and ribosomes, they leave nucleus. mRNA carries genetic information to ribosomes. Ribosome is a site, the site for protein synthesis, right? For protein synthesis, can see that. Substances entering. Now, proteins are enzymes that are needed to make ribosomes, okay? Nucleotides, building blocks for DNA and RNA, they enter. ATP provides energy, so they enter the nucleus, and some hormones. So chromosomes contains hereditary material DNA that is organized into genes, which controls the activity of cells and inheritance. You can see we've talked about that. All right, so you can see nucleolus 0.2 to 0.5, one or more found nucleoli containing DNA and RNA functioning to make ribosome. So that's about nucleus, guys. All right, let's go. So knowledge check. Okay, which part of the cell is often continuous with rough endoplasmic reticulum? You should be able to get this. Okay, right? Cell surface membrane, no. Golgi body, no. Mitochondria, no. It is nuclear envelope. So the answer is delta. D is the answer. Let's go. Now, we're talking about endoplasmic reticulum. Guys, remember that endoplasmic reticulum, all right, the nuclear membrane is continuous with endoplasmic reticulum. So endoplasmic reticulum actually, they're usually membranes, all right, which surround an enclosed inner cavity. So this enclosed inner cavity, you can see that, is called cisterna. This enclosed inner cavity is called cisterna space. Okay, right? So they are kind of interconnected membrane system. So if you check, there are two types. This one has ribosome on the outer membrane. All right, it is called rough endoplasmic reticulum. You can see this. This does not have, all right, ribosome on the outer membrane. It's called smooth, no and ribosome. Guys, I want you to look at them critically. Remember, they are membrane, right? Systems, interconnected membrane system. But you could be asked to differentiate between this and this. Now, if you look at the rough endoplasmic reticulum, of course, number one difference is that it has what? Ribosome. This doesn't have ribosome. Okay. Number two is that, all right, it's made up of flattened membrane sac. Can you see it's flattened? Now that's for rough. Rough is flattened membrane sac. Smooth is cylindrical membrane. Okay, can you see it's cylindrical? Yeah. So now another thing is that this rough endoplasmic reticulum is continuous with nuclear membrane. Okay. Now, smooth is not continuous with nuclear membrane. I've mentioned three differences. One has ribosome, that's rough. One doesn't have ribosome, okay, and all of that. One is flattened, one is cylindrical, that's smooth. Okay, this is continuous with rough nuclear membrane. This is not. Now, if you check nuclear membrane, smooth endoplasmic reticulum is a site for synthesis, all right, and transport of lipids and steroids. Okay, so now organs where we have a lot of lipids and steroids, you find more of this there. For example, organs like testes, ovaries, they make what we call steroid hormones. Okay, so steroid is a part of lipid. Okay, so you find more of this plenty in such tissues and organs. All right, but any cell that makes a lot of protein will have more of rough endoplasmic reticulum. Is that okay? Because the function is that it synthesizes, modifies, and transports protein. That's rough endoplasmic reticulum. Okay, so let's look at the next. So you see rough endoplasmic reticulum, an extensive membrane system with ATS. So the type of ribosome there is ATS, not 70, right? are sites for protein synthesis which produce the rough appearance. Yeah, the RER provides a pathway for transport of materials through cells, forms a complex 3D system of sheet-like membranes and tubes enclosing fluid-filled sacs. Okay, proteins made by ribosomes on RER enter sacs and move through them. Transport vesicles bud off, all right, from RER and join, forming the Golgi body. So you see, Golgi body is continuous with rough endoplasmic reticulum. So smooth endoplasmic reticulum site for lipid synthesis and steroids, cholesterol and reproductive hormones. You see, all right. So you find them there in a lot. Okay, in reproductive organs. Meshock or tubular, now you see that it's tubular or cylindrical membrane vesicles with fluid-filled sacs that have no ribosome on its surface, guys. So now I want to just show you something. Okay, so if you check this, we did not talk about their similarities. Both of them are made up of, of course, a membrane system, interconnected membrane system, both smooth and rough. So this is rough, guys, right? Don't forget that the rough has ribosome attached to the outer membrane, and at the type of ribosome here is ATS ribosome. That's the type found on rough, guys. Now, I'm not going to draw so much of that. Now, this is smooth, right? Very smooth, very, very smooth, guys. Watch that. Okay. Now, I want you to see. Okay, there's no ribosome attached there, but there's something they have in common, right? Both of them form vesicles. These vesicles are for transport. For example, in rough, okay, rough transports proteins. Now they both transport protein to Golgi for modification. But they both transport materials to Golgi for modification. While rough transports protein, smooth transports steroids and lipids for modification, guys. So the first thing they have in common is that they are made up of interconnected membrane, all right, system. This interconnected. The second is that they both form vesicles for transport, and the third one is that they both enclose. They're made up of membranes which enclose an inner, all right, cavity. So this is cisterna. They both have cisterna, right? Cisterna is found here. Okay, let's go to the next thing. So now we come to Golgi. Don't forget, guys, that Golgi, all right, is continuous with rough endoplasmic reticulum or endoplasmic reticulum. So that's the Golgi, guys. Okay, so the Golgi actually made up of membranes. Okay, membrane sacs. These are membrane sacs. Okay, which are arranged as a flattened sac. Okay, the membrane sacs arranged as flattened sacs. So you can see one thing about them is that they could be curved, okay, or parallel, right? But if you look at Golgi, it has some distinct features. For example, it has bulging ends. You can see bulging ends here, bulging ends. Okay, now bulging ends here. Now the first part here is called cisterna. Guys, don't forget cisterna. Yeah, cisterna. No, the cis end. Sorry, I wanted to say that because Golgi came from endoplasmic reticulum, so he also has cisterna. So this end here is called cis end, and we have trans end. All right. So now trans end and cis end. Now, the trans end is where modified vesicles carrying modified products bud off or pinch off. Okay, vesicles are always pinching off from here for transport outside the cell or inside the cell. Okay, but the cis end here, vesicles from endoplasmic reticulum come here to fuse. They fuse at the cis end, right? But they bud off at the trans end. Okay. So when they fuse here, they pass through the cisterna for modification. After modification, right, see them budding off or pinching off. Okay, so you see what we have here. So Golgi flattened, stack of flattened membranes enclosing hollow sac cisterna formed by transport vesicles which bud off the RNA and make protein and are broken down to form Golgi vesicles. So these vesicles are called Golgi vesicles, all right? So collect, processes, so that's the function. Collect, processes, modifies, and sorts molecules that are ready for transporting Golgi vesicles to other part of the cell or out of the cell. Now, secretion, exocytosis, fusion of vesicles with plasma membrane to release content. Now, what this means is that, so Golgi helps to modify. It does a lot of modification, okay? When it's done with modification, it packages it in vesicles and transports it. Now, I want you to know that Golgi modifies protein. One of the things it modifies is protein. Let me call protein P, all right? It modifies protein, okay? So how does it modify protein? One of the ways it modifies protein is by folding. So it can fold proteins, right, to form different levels of protein structure. Okay, now folding or coiling protein. So protein can be folded, and then that gives us the secondary, right, structure, level of protein structure. Tertiary, okay, can also give us tertiary level of protein structure. I call it TET. It can as well give us the quaternary. So this is one of the ways Golgi modifies protein. Is that okay? Yeah, that's one of the ways. It can give us these three levels: the secondary structure, a level of protein structure, of course, quaternary, tertiary, and quaternary. Okay, it can also add prosthetic group. Okay, like if this is hemoglobin, it can add the hem group. Now, the one way it modifies P, which is protein, is by folding it, producing these structures. Another way Golgi modifies protein, this is very important, is by addition of glucose. I'll call glucose G. So it's a P plus glucose. Okay, it can add glucose to it, and that process is called glycosylation. Glycosylation is spelled Glysylaton, glycosylation. So when glucose G is added to protein, it gives us glycoprotein, and this occurs in Golgi. So glycoprotein is made in Golgi, guys. Now, one of the examples of glycoprotein, guys, you will never know. All right, it will shock you, guys. One of the examples of glycoprotein is what we call mucus. Mucus, that yellow fluid or milky fluid that you blow out of your nose, okay, in dusty environment, that usually trap pathogens. So mucus, okay, is a glycoprotein. So if we're looking at how glycoprotein is made, remember that it's going to involve protein. That means that, remember, if you're going to produce glycoprotein, the first thing that happens is transcription, because protein synthesis, all right, starts with transcription, right? And then transcription is synthesis of messenger RNA, and then after transcription, which occurs in nucleus. So nucleus is involved, then it's taken to ribosome for translation. Ribosome can be found floating on the cell, on the cytoplasm, or attached to RER. Okay, so now in ribosome, the mRNA is translated to form protein. Then the protein is transported to this guy here, Golgi here, at this end. When it fuses here, the protein undergoes glycosylation, where glucose is attached to it to form glycoprotein. Okay, and then once it's formed, the next thing that happens is that the glycoprotein is packaged in vesicles. You can see these vesicles, packaged in vesicles, budding off at this trans end. So the vesicles carrying mucus or glycoprotein buds off from the Golgi, and when it buds off from the Golgi, it moves, all right, across the cytoplasm, guys. Don't forget, this is division of labor. Everybody's involved in this game, right? So you see the vesicle carrying this moves across the vesicle, so across the cytoplasm, right? So the question is, what makes it to move? What makes it to move is cytoskeleton and microtubules. They contract and push these vesicles carrying mucus, right? Because this mucus is going out of the cell, okay? And then when it moves across the cytoplasm, it moves towards the cell membrane, fuses with it. All right, don't forget that movement requires ATP. So that means mitochondrion is also involved. It provides ATP for the movement, for the budding of, for the formation of vesicles. Okay, now when it fuses with the membrane and then it is released. Okay, so you can see that This is called exocytosis. So the vesicles fuse with the cell surface membrane, and then the mucus is released. All right, guys, you can see that. So guys, don't forget this division of labor. Okay, so what are the organelles involved? Nucleus is involved for transcription, ribosome or RER is involved for translation, Golgi is involved for modification, all right, packaging, and then transport. Of course, cytoskeleton move it, and then that provides energy. What provides energy is mitochondria. Cell membrane is involved, then there it is. So you see that in cells such as Golgi, no, not Golgi, goblet cells. Yeah, goblet cells. Goblet cells synthesize and secrete mucus. So they're going to have large number of Golgi. So all secretory cells have large number of this single membrane bound. Remember that Golgi is single membrane bound. Okay, endoplasmic reticulum is single membrane bound. Okay, so it will have a lot of these. All right, Golgi. Yeah, so see that. Okay, you can see that also Golgi forms vesicles. All right, some of these vesicles formed by Golgi, they're called lysosomes. Lysosomes board off. Okay, vesicles, large vesicles are lysosomes, but lysosomes carry digestive enzymes. Not all vesicles are lysosomes. The ones that carry digestive enzymes are called lysosomes. Okay, so but you see, vesicles can also carry glycoproteins, can also carry, all right, glycolipid. Don't forget that vesicles from smooth endoplasmic reticulum also come to Golgi because they are carrying lipid. And then when lipid comes to Golgi, let me call lipid L. LP is lipid. Is that okay? So this LP can be modified by adding glucose G. So that's glycosylation, and that gives us glycolipid. So glycolipid is made in Golgi, right? When Golgi adds glucose to the lipid, all right, it forms glycolipid, all right, that's glycosylation. Now this can also modify LP, which is lipid, can also be modified by addition of phosphate group. So Golgi can add phosphate group to lipid. Addition of phosphate group is called phosphorylation. So lipid can be phosphorylated in the Golgi to form phospholipid, which is a core structure of cell membrane. So that's phospholipid. Is that okay? So guys, don't forget that phospholipid. All right. So you see, phospholipid, phospholipid, phospholipid. Where are you? Okay, that's it. Okay. So Golgi can make lysosome. Now that is about Golgi. So let's go to mitochondria, guys. So that's the powerhouse of the cell. Okay. So but I want to show something. The new forming vesicles are budding off here. They're fusing here, budding off. Okay. So mitochondrion, 1 micrometers. Nucleus is about 10 micrometers. So mitochondrion has, there are 3 things it has. Okay, it has double membrane. It has an envelope. Okay, provides energy, for synthesizing lipids more in areas that require maximum energy. So all the cells that require a lot of energy will have mitochondria. Okay, but you see, like cilia, okay, or muscle cells. So mitochondria is a powerhouse. Let's talk about this guy, but let's do knowledge check. So which animal cells would have the most extensive Golgi bodies? So which of them is secretory? Pause the video. Which of them secrete something? Okay, because Golgi bodies are found in secretory cells. So which of them? Pause the video and answer it. A says ciliated epithelium cells. Now, ciliated does not secrete, it just moves the mucus. Goblet cells, yeah, goblet cells secrete mucus. It will have most extensive Golgi bodies. Okay, red blood cells does not secrete, transport oxygen and CO2. Smooth muscles contract, so it will have more of mitochondria. Okay, this will have more of Golgi body. All right, so the answer is bravo. Let's go, guys. Under knowledge check. Yeah, let's look at what happens during mucus secretion. I want you to pause this video and try the answer. Let's check whether you are learning. Okay, please make sure you are dropping comments, all right, on our YouTube channel. Tell us what you're learning. Look at it. When mucus is secreted from goblet cells, these events take place. The first one is addition of carbohydrate to protein, fusion of the vesicles with Golgi, with cell surface membrane, secretion of glycoprotein. All right, and then separation of vesicles from the Golgi body. What is the sequence of this event, guys? What is the sequence? What is your answer, guys? All right, let me paint a picture. I did this before. Okay, now this is flattened membranous sac. Okay, and that's Golgi. So what happens is that Golgi packages, first of all, it modifies our glycoproteins by adding glucose to it through a process called glycosylation, packages it, all right, in vesicles. Oh, pardon me. So these are vesicles. So the vesicles move across the cytoplasm. Remember, this is a cell. Probably, you know, what type of cell is that? Goblet cells. So these vesicles move across cytoplasm by the help of microtubules, okay, using ATP, and then fuses with it. Once it fuses with cell membrane, and then it releases it. So which process should come first, guys? All right. So first of all, is the addition of carbohydrate to protein. That's glycosylation, and after that, it's going to burn off. So what's next? So one is there. So one starts it. That means C and D out. Okay. So after addition of carbohydrate, there's a fusion, no, secretion, no. Now separation of vesicles from Golgi. Can you see the vesicles start separating from Golgi? So after one is what? 4, 1, 4. Okay. After separate, they will move towards cell surface membrane. So 2, fusion of vesicles, cells. Yeah, that's what happens. 2 is next, and then 3 says secretion. Yeah, secretion. So guys, that is okay. So look at this. It's not a big deal. So what is happening here is addition. Addition happens, and this is number 1. Okay, 4 is separation. This is number 4. 4 is okay, separating, and this is 3. They are fusing with the same surface membrane, right? Is that 3 or 2? That's 2, fusing. Okay, and 3 is that they are secreting. Okay, so 1 first, and then after 1 is 4. 1, 4, 2, 3. That's A. Okay, back to this guy, the powerhouse of the cell, mitochondrion. Guys, don't forget mitochondrion. Look at this guy. It powers all cell activity, right? Energy source. Now look at mitochondria. Number 1, it has double membrane. Don't forget that. It has double membrane. But can you tell me what structure is this surrounding mitochondria? Can you see? This looks like endoplasmic reticulum. Maybe like rough. Okay, but let's get back to mitochondria. So mitochondria, see, the first structure is double membrane. Remember, nucleus also has double membrane, but it has also folded inner membrane called cristae. This folded membrane is called cristae. All right, it has double membrane, outer and inner. The inner is folded to form cristae. Cristae provides large surface area for enzyme action during photosynthesis. So cristae is a site of oxidative phosphorylation. Right now, this is the matrix, the inner compartment matrix. You can see the matrix here. The matrix have circular DNA. So mitochondria has circular DNA. All right, just like prokaryotes, it has circular DNA, just like chloroplast. Chloroplast has circular DNA, right? Mitochondria has 70S ribosome, just like chloroplast and prokaryotic cell. They have 3 things in common. They have circular DNA, okay? They have 70S ribosome. Of course, this time around, the double membrane is shared between mitochondria and chloroplast, okay? Yeah, so you can see that, okay? So if you check, it was told that mitochondria and chloroplast, they actually evolved from a prokaryote because they have 70S ribosome and circular DNA. But now they don't have cell wall again. They have lost cell wall. We're going to discuss that later. But look at it. It has a matrix that contains 70S. So the type of ribosome here is 70S, but the type of ribosome on endoplasmic reticulum is 80S. They are different. 70 is smaller, 80 is bigger. They do the same function. All right. 70S is found inside chloroplast and mitochondrion here, which is used to make some of the mitochondria's own protein. So mitochondria makes its own protein. So mitochondria has circular DNA. It has DNA. It contains information required to make proteins required for respiration. So mitochondria has DNA. Now, because it has DNA, it will have other nucleic acids. It will have mRNA. When DNA is transcribed, it forms mRNA. Is that okay? So it's going to have mRNA here. It's going to have transfer RNA here. It's going to have, because it has 70S ribosomes, so you have ribosomal RNA. Guys, don't forget this. Okay? So transcription occurs, replication occurs here. Translation also occurs here. Okay? All right. Crystal folding of inner membrane that projects into the interior solution matrix, interior solution matrix. All right. Intermembrane space between the two membranes. Porine transport proteins in outer membrane forms wide aqueous channels, allowing water-soluble molecules from cytoplasm to intermembrane space. Inner membrane, selectively permeable, controlling entrance of ions and molecules into the matrix. So inner membrane helps to set up proton gradients. We'll do that in respiration. Is that okay? So don't forget what I've said here. Let's go, guys. Yeah. See, this is circular DNA. That's ribosome. Okay. See this matrix. That's the outer membrane, inner membrane folding. Okay. See, folding. All right. Inner membrane helps to maintain proton gradient. Okay. So that's sorry, intermembrane space. Inner membrane is folded. Okay. So look at it. So the function of mitochondria, major function is ATP synthesis, right? Mitochondrion has four things in common with chloroplast. Number one, they both have double membrane. Number two, they both have 70S ribosome. Number three, they both have circular DNA. Number four, they both make ATP. Number five, what's number five? Okay, what is number five? Tell me, what do you think number five is? Is it number 5 or 4? Okay, let's leave it at 4. All right, so let's discuss the fifth one later. So knowledge check: which cell structures produce ATP? I gave you the answer. So tell me the answer. See, which cell structures produce ATP? Chloroplast, mitochondria, 2 of them produce ATP. So the answer is what? B. Nucleus doesn't produce ATP. Nucleus carries out transcription, okay, controls cell activity by transcribing genetic information to mRNA, okay. So it's 1 and 2. So let's go. The next knowledge check: which cell structures are required for the formation of lysosomes and hydrolytic enzymes that they contain? Remember that hydrolytic enzymes are proteins. So which one makes protein? Pause the video. Tell me the answer. You can tell me the answer, and if you get the answer, we'll send you chocolate. Okay. So don't forget, enzymes are proteins, and they are synthesized. Proteins are made with rough endoplasmic reticulum. So rough is part of it. Okay. Remember that formation of lysosomes. Lysosomes are formed by Golgi. Golgi modifies the protein. Okay. Mitochondria provides energy for both the synthesis of enzyme, the modification, the formation of vesicles, the transport. It powers everything. So all of them are involved. So the intercept is B. That's the answer. All right, knowledge check, guys. Which cell structures contain rRNA? rRNA is ribosomal RNA. Is found in ribosome. What's the answer? Okay. Pause the video and tell me the answer. So the question is, where do we find ribosome? Anywhere we find ribosome, find rRNA. Okay. Now, ribosome, remember I told you chloroplast contains 70S ribosomes, so it contains RNA. Mitochondria contains 70S ribosomes, contains rRNA. Nucleus contains nucleolus. Nucleolus synthesizes RNA. Synthesizes ribosome, so it has rRNA. So it's found here. Smooth endoplasmic reticulum doesn't contain it, right? So our answer is 1, 2, and 3. Bravo. Okay. Yeah. Next, check. Mitochondria are thought to have evolved from prokaryotic cells that were ingested by an ancestral cell. Okay. So yeah, that's called endosymbiont. We'll discuss that. Which feature have prokaryotes lost during their evolution into mitochondria? Now, prokaryotes have cell wall. But pause the video, okay, and solve the question. So the question is, if mitochondria came from prokaryotes, that means they're involved. So what are the features that prokaryotes lost? What are the features that are in prokaryotes, but they are no longer in mitochondria? Let's check. Yeah, prokaryotes have cell wall, mitochondria don't have cell wall. So that's the answer. But why is it wrong? Because prokaryotes have circular DNA and mitochondria has it. Okay? Yeah, but none of them has endoplasmic reticulum. So this is out of it. Both of them have ribosome. So the only one prokaryote lost was cell wall. So the answer is A, alpha. Yeah, guys, look at this. I want you to solve this. Pause the video and try to solve this question. So the diagram shows a typical animal cell as seen using an electron microscope. Okay, which of the numbered structures are needed for protein to be secreted at the cell surface membrane? Okay, for protein to be secreted, just for it to be secreted, right? So which one is needed? Now, when proteins are made by the rough endoplasmic reticulum, if it is a synthesis, rough endoplasmic will have been involved. Rough contains ribosome, so no, this structure 2 is ribosome on rough. Okay, this 5 looks like lysosomes. Okay, yeah, 3 is mitochondria. What is 4? Smooth endoplasmic reticulum synthesizing lipids and steroids. So enzymes are proteins, so this cannot be there. Okay, yeah. Now for secretion, Golgi packages in vesicles, so Golgi must be there, right? And then what else is involved? Okay, of course, mitochondria provides energy to form vesicles. The vesicles moving and then fusing. Okay, so mitochondria involved. Remember, we're dealing with proteins to be secreted. So 1 and 3, they are involved in the secretion. Okay, the answer is C. Golgi is involved. It forms vesicles that packages the enzyme. Mitochondria provides the energy for the formation of the vesicles, the movement, and the exocytosis. Okay, if they had mentioned synthesis, probably would have included 2, but just said secretion, secrete. So the answer is Charlie, C. Golgi and mitochondria. C is the answer. All right. Where are cisterna found in a cell? Yeah, pause the video. Cisternas, they are what? What are cisterna, guys? They are what? Okay, an enclosed inner cavity. So cisterna, is it found in endoplasmic reticulum? Yeah. Okay, don't forget, endoplasmic reticulum have this cisterna, right? Whether it is rough or smooth, that enclosed inner cavity cisterna. Remember, endoplasmic reticulum forms is continuous with Golgi. Yeah, Golgi has it too. Mitochondria doesn't have it. Okay. All right. So the answer is 1 and 2. Okay. So that is A. All right. Good. Look at this, guys. So this is knowledge check, and that knowledge check very important, guys. Knowledge check. All right. So knowledge check. Watch this. It says 6 organelles. Yeah. Let's go. Let's go. Okay. 6 organelles found in eukaryotic cells are shown. So eukaryotic because it has nuclear membrane. This is number 1 is what? Pause this video and solve it, people. So number 1 is what? It is, of course, nucleus. This is nucleolus. These are chromatin. They are condensed to form chromosomes. 2 is mitochondrion. 3 is centrosome. Central. 6 is what? Golgi body. 4 is what? Rough endoplasmic reticulum. 5 is what? Chloroplast. Okay. Now solve the question. Which organelles are involved in the synthesis and secretion of glycoprotein? This is very interesting. Remember glycoprotein. Okay. Where is protein made? Protein is made in Golgi. Good. Where is it modified? By addition of glucose chain, carbohydrate chain. Okay. Golgi. So 4 and 6 are involved. But for protein to be made, all right, the information from for making DNA is stored where? In nucleus. So that information is transcribed in the nucleus to mRNA. mRNA comes to number 4 ribosome to be translated to form the protein. The protein is transported to Golgi, okay, for modification. So and then mitochondrion powers all these processes, powers transcription, powers transport and synthesis. Of course, protein, it powers the modification. So it's going to involve 1, 2, 4, and 6. So 1, 2, 4, and 6, that's B. The answer is B, guys. So our answer is B. Bravo is the answer. Okay. Thank you for watching. Kindly watch out for our next episode. Okay. Leave your questions at the comment section. Please like, subscribe, and turn on the notification button on 13 Universal YouTube channel for prompt notification on our new videos. Remember, this is Shedro. Thank you for hanging around. Bye. See you next episode. Bye.